Without further ado, Stephen Altschul.
Thank you for coming to the second of these talks. I will be speaking today mainly about protein sequence alignment, although a lot of what I say is applicable to DNA sequence alignment, and I'll have some things to say about that towards the end of the talk. Now, last week, just to plunge in, we discussed how scoring systems for comparing two protein sequences can be expressed in terms of a square matrix with the scores always given in this form. So basically any scoring matrix that one is using for local pairwise alignment takes this form. The log of the base of the logarithm is arbitrary, and if one uses log to the base 2, the scores are said to be expressed in bits. We will generally be talking about bit scores today. Now, what we didn't discuss last time is where the target frequencies for scoring systems come from. And our central question today is, what is the optimal way to choose these target frequencies that correspond to the scores? For many years, people tried a large variety of scoring systems constructed using a lot of different rationales, ranging originally from match-mismatch scores to scores based on the genetic code to scores based on multidimensional scaling, on various properties of amino acids. But what one learned is that all scoring systems are implicitly log-odd scoring systems, and so that choosing any of these other scores was choosing a set of target frequencies. And the one set of scores that did this explicitly for a long period of time were scores constructed by Margaret Dayhoff called PAM scores, and these seemed to work better than most other scores, and we've subsequently learned why that was. But we will get to the PAM scoring system shortly. So to understand why one wants to choose a particular set of target frequencies or how to choose it, I want to consider a schematic histogram of a database search. So the way to understand this graph is that on the x-axis here we have the normalized score that we talked about last time, and here we have the number of local alignments we are finding with that score. And as we said last time, purely by chance, the number of alignments that you find at a given score tends to decrease exponentially. And here, at basically the log of the search space size, which is the product of the length of the query times the length of the database and residues, is where you expect on average to find one random alignment with this score or greater. And that is completely independent of what the scoring system is or what your target frequencies are. Now, in this schematic diagram, I have represented by X's local alignments that do not represent any true biological relationship. But the reason one does these database searches is that frequently your query sequence will have a segment that is related to segments in real database sequences. And these alignments will get high scores that frequently, in fact quite generally, will be much higher than the score you expect by chance. And so the red pound signs here represent the scores for related alignments. And typically you will have many more scores way off to the right that I'm not showing here. Now, the reason one, of course, if one knows ahead of time, as this slide implies, which are related and which aren't, there isn't a reason to do the database search. In general, one does not know which sequences are related and which are not. And therefore, what one wants is to have a scoring system that will pull the scores of related alignments as far to the right, give them the highest score possible, because the noise is always going to be there. The noise will be there no matter what scoring system you use. And so the question is, what target frequencies will maximize the scores for related alignments? Now, I won't prove the answer to this, but the answer is quite simple and is sort of a beautiful result. And that is, if we can characterize the true alignments by the frequencies with which they align pairs of sequences. So if we take all true alignments and we look at the pairs of amino acids that are aligned in the true alignments, and if we characterize the frequencies with which pairs of amino acids are aligned by Q sub ij, then if we use those target frequencies to construct scores, those scores will maximize, on average, the scores of the true alignments. So in other words, just to say this again, the scores that one wants to use to maximize the scores of true alignments are one where the target frequencies characterize the frequencies with which pairs of letters are aligned in the true alignments. Now, when one looks at this score, this actually makes beautiful sense. What this says is that if you are seeing a given pair of amino acids aligned in true alignments more frequently than they would occur by chance, because this is the probability that you would align these two letters by chance, you then give that pair of letters a positive score because this ratio is greater than 1 and the log of a number greater than 1 is positive. If you were seeing a pair of amino acids aligned less frequently than chance, you get a negative score. And if there is really no difference in true alignments and chance alignments with which a pair of letters is aligned, the score is 0. So again, these scores make beautiful sense. Now, one thing that I have skipped over here is that to say that there is a set of target frequencies that characterizes true alignments is sort of an incoherent concept unless we add something to it. And to understand why this concept alone doesn't make a lot of sense, I want to look at some alignments that represent true relationships. These are 4 alignments, all of the human beta globin sequence. The beta globin sequence is written along the top here, and I've repeated it 4 times. Here, I've aligned the human beta globin sequence with the beta globin from a ring-tailed lemur, and here's a picture of a ring-tailed lemur. On the center line, we have echoed the identical amino acids, and so wherever you see a letter on the central line, it means the 2 sequences have the same amino acid. Here I've done the same thing for an alignment to the beta globin from goldfish, and if you look carefully, you'll see that in fact the goldfish has 1 extra amino acid, and I've had to put in a gap character here in order to get these sequences to line up as well as possible. This is a globin from a bloodworm, My understanding is that bloodworms do not have blood, but they eat blood. They nevertheless have proteins that need to bind oxygen, and so they have globins that do that. And even soybeans, which are diverged from humans by more than, I think, about 1.2 billion years, they have molecules that also bind oxygen. They certainly don't have blood, but they have molecules that bind oxygen and are related to the globins in humans. And now going back to the ring-tailed lemur, or even to a different species, if I were to show you the beta globin from a chimpanzee aligned with the human, and we're talking here about the most frequent alleles in these species because sometimes you have slight variations, but if you look at the most frequent alleles, human and chimpanzee beta globin are identical. There's no difference whatsoever in their amino acid sequence. With gorillas, I think there are two changes, and by the time you get to ring-tailed lemurs, which I think diverged somewhere around 50 million years ago, you have accumulated many more changes. Now, if you take this sequence and others, say from other alignments, to estimate the frequency with which various amino acids are aligned, you can do that by just counting up the occurrences. So let's say we wanted to see how frequently valine was aligned with valine. Well, here is one occurrence, 1, 2, 3, 4, here's another, here's another. So one can go through this alignment and many similar ones and say how frequently in all alignment positions does one see valine aligned with valine. Now the problem is that if you did the same thing for the human versus goldfish alignment, you would find a different set of frequencies. So although there's one more valine aligned with valine here at the start, this valine is aligned with an isoleucine. This valine here is aligned with a leucine, and this one is aligned with an isoleucine, and so forth. Here's one that is, in fact, aligned with a valine, but one would find that the frequency of valine aligned with valine is a lot lower when one gets to this more distant relationship. One can then go on to bloodworm and find that there are even fewer valines aligned with valines. And down here, I think we have only these two here at the beginning, as far as I can tell. So the point is that the frequencies that various amino acids are aligned in true alignments depends on how closely the sequences are related. If you have two very closely related sequences, then most amino acids will be aligned with the identical amino acid, and the great bulk of the probability will be on the main diagonal. That is where the amino acids aligned with one another, if you put them in a square matrix, occur. As you go to more distantly related true relationships, you have less of the probability on the main diagonal. There are many fewer identities when you are looking at distantly related sequences. So although the idea of there being a single set of target frequencies that are optimal is not a coherent concept, one might very well come up with a concept of a series of different target frequencies with different associated scoring systems that are tailored to different degrees of evolutionary divergence. Now, let me go back to Margaret Dayhoff's PAM model of protein evolution because this idea was one that she had very early on. And the way the PAM model was constructed, let me explain how this works. Basically, she decided to come up with a Markov model of protein evolution. And the idea is that during a given period of time, amino acid I has a certain probability of mutating into amino acid J. Now, the idea of mutating here is a little bit more complicated than simply the mutation taking place, which is would happen. We're talking here about point mutations, which are mutations at single positions of proteins, and a point mutation will change one amino acid to another. It occurs because you have a mutation at the DNA level, and the protein that it is coding for then is changed as well, although there can be mutations at the DNA level that are silent that do not change the amino acid. Now, not all mutations get accepted by evolution. So there are many mutations that are selected out of the population because they cause the organism to be less fit. They may even kill it immediately if it damages the protein sufficiently. And so only certain mutations get accepted by evolution, and these are called accepted point mutations. Now, Dayhoff, I guess, wanted to call her model the accepted point mutation model of evolution, but APM was hard to pronounce, and so she switched the first two words, and it became the point accepted mutation model of protein evolution, or the PAM model of evolution. Now, one PAM in her model corresponds to a single accepted substitution on average per 100 amino acids. So if you have a protein of length 100, on average, you're expecting a single one of them to mutate and be accepted by evolution, and that is what is called one PAM. Now, the problem is that no one tells us what these probabilities are that one amino acid is going to mutate into another. And so the only way we can tell this is by looking at some data, and the data that we look at are alignments. And so there is a certain circularity here in that we want to construct scoring systems for making alignments, but we are going to use alignments to construct the scoring systems. Now, one can break out of this circularity if one goes back here, in that it doesn't really matter pretty much what scoring system we use here. We are going to construct this alignment very easily. There are no gaps. It's very obvious how to line this up. And so we can look at an alignment like this and see how frequently various amino acids change into other ones. From an alignment like this, you can't really tell which direction the change has taken place, but one can kind of count it in both directions, or one can look at other organisms and infer what the ancestral amino acid might have been. Now, what Dayhoff did was look only at proteins that are greater than 85% identical. So there's only about one change every 7 positions. It's very obvious how to line those proteins up properly and to estimate these frequencies. And so that is what she did. And given these probabilities of one amino acid mutating into another, for one PAM, one may infer, if one assumes this process is a uniform process over time, one may infer these probabilities for any PAM distance by multiplying this matrix by itself to get 2 PAMs, multiply the matrix again to get 3 PAMs, and so forth. And so one may construct these mutation probabilities for any PAM distance, and one can therefore estimate the probability of seeing a particular pair of amino acids. A probability of seeing amino acids I and J lined up with one another are the probability that you start with amino acid I, and after a certain number of PAMs that has changed into J. Now, of course, what we are seeing are generally 2 sequences in the current day, and so to relate one of the sequences to the other, you have to simply go back along the evolutionary tree to their ancestor and then forward along the evolutionary tree in the other direction. But if one assumes that there's no particular direction to the frequency with which things mutate, one can do that and basically assume that you're starting with the amino acid you see in one sequence, and the total evolution you're talking about is the total time it takes to connect it to the other one. Now, the substitution scores that you get from the PAM model are then simply constructed this way. You estimate the frequency with which the pairs are aligned, and you get the log odds scores. You can write it this way as well, and it turns out that by the way she constructed these probabilities, that although these probabilities are asymmetric, the frequency with which I mutates to J is not the same as the frequency with which J mutates to I, the target frequencies and the scores, by the way she constructed these frequencies, are in fact symmetric. Now, one should say that 100 PAMs, just to make this clear, 100 PAMs is the total amount of evolution where there have been 100 changes on average over a protein of length 100. But some of those changes may have taken place in the same position. So a given position may mutate multiple times, and in fact, it may mutate back to what it started as. And so using this model, one can calculate that 100 PAMs of evolution, the sequences are still actually 43% identical, because many positions haven't changed at all in that time, and some positions have changed multiple times but have changed back. And 250 PAMs corresponds to proteins that are about 20% identical. I mentioned the 250 PAMs because when Dayhoff first published her scoring systems, there were very few proteins to work with. She was comparing proteins that were globally related, and she found that 250 PAMs was a good set of scores to work with, where she was looking for very distantly related proteins. For a long time, these were the only scores that people used, and people sort of forgot that there were, in fact, PAM scores for any given PAM distance. But we will discuss a variety of different PAM distances today. Now, a more recent approach to constructing scores was taken by Stephen Georgia-Henikoff. Last time I showed you the BLOSUM 62 matrix, which they constructed, and they also construct their matrices explicitly as log odds matrices. But rather than trying to extrapolate what the frequencies for aligned pairs of amino acids will be from a mutation model constructed from very closely related sequences, the Henikoff said, well, let's go and look directly at distantly related sequences. The problem with doing this is, how do we know that we have gotten the alignments correct? Dayhoff can very easily get accurate alignments if you're dealing with proteins that are at least 85% identical. If you're dealing with proteins that are 50, 40, 30% identical, it is a lot harder to get the alignments correct. And so their way of doing this is to look at multiple alignments where you have many sequences from a protein family, and from multiple alignments, even distantly related sequences, you can find accurate alignments of blocks, little regions in these proteins that are related across all of them, and they constructed their target frequencies from looking at these blocks. To get various degrees of divergence, they would cluster or group sequences that are more than a certain percent identical, and they would count changes only between these groups and not within them. And so the BLOSUM 62 matrix refers to taking their block database of blocks, grouping sequences that are 62 or greater percent identical, and then only looking between the blocks. One thing that is a little bit confusing, if you're don't become aware of this early on, is that the numbers in the PAM and BLOSUM series, they have very different meanings, and they sort of go in the opposite directions. And so a large PAM number corresponds to proteins that are greatly diverged, whereas a large BLOSUM number tends to be associated with sequences that are not very greatly diverged. And if you think about where this number comes from, you'll see why that is. Now, at this point, I want to introduce a new concept, which is called the relative entropy of a matrix. Now, don't be frightened off by the name. It actually corresponds to a very simple concept, and that is, if we have two sequences that are related, and let's say we are using the PAM model, and we have two sequences that are, say, 100 PAMs diverged, and we have lined these up accurately with one another, we can ask, what is the average score for this true alignment of related sequences? Well, it's easy to calculate that because we know the PAM 100 scores. We have that for things that are diverged by 100 PAMs, but we know the frequency with which those scores occur because the target frequencies tell us how frequently various pairs of amino acids are aligned. And so by simply taking all the scores and averaging them by the frequency with which they occur, you get this quantity. If we expand this out, it looks like this. And this is a quantity which is called the relative entropy in information theory. It is a very important concept in information theory, but for our own purposes here, one can simply think of this as the average score, or let me even put it this way, the average amount of information that one gets per alignment position. If the scores are expressed in bits, then the relative entropy also has units of bits, and one can simply think of this as saying, how much information do I get per position to show that two sequences are actually related? This sum, by the way, looks somewhat similar to a quantity we looked at last week, which was the average score for random sequences. There, rather than Q sub ij, you had Pi Pj, which is the frequency with which a score occurs if you just pick two random amino acids. And that quantity, in fact, is a negative number. In fact, we required it to be a negative number. The relative entropy, on the contrary, is a positive number, and it is the average score for related sequences as opposed to unrelated sequences. Now, one can graph this relative entropy as a function of PAM distance, because since we can calculate the frequency with which pairs of amino acids are aligned at different PAM distances and get the different scores at different PAM distances, we can calculate the relative entropy. And in this graph, this curve is the relative entropy as a function of PAM distance. So at 0 PAMs, zero PAMs is simply two sequences that have not diverged at all. Therefore, all of the probability is on the main diagonal. You have 0 probability of two amino acids that are not identical being aligned. In other words, all the scores off the main diagonal are the log of 0 or negative infinity. So a PAM 0 matrix is simply positive scores along the main diagonal with no mismatches allowed. The relative entropy is slightly over 4 bits for proteins. As you get out to 100 PAMs, one has dropped to somewhere around, I think it's around 1.2 bits. Here at about 120 PAMs, you're getting 1 bit. And out at 250 PAMs, you're down here at somewhere around 1/3 of a bit per position. Again, this makes very good sense that the amount of information that you have to say, are these sequences related, decays over time. If one goes back to these alignments, you don't have to look at much of this alignment to realize this could not have occurred by chance. There's a lot of information that this is related. Whereas this alignment, in fact, may very well, you may very well see something like this by chance in a database search. It is not at all obvious simply by looking at this alignment alone that these sequences are related. So the amount of information decays over time. Now, the interesting thing is that we said that in a database search of a given size, you will need a certain amount of information to rise above background noise. And so let us imagine we are doing a database search. These days, it might be a relatively small database search. You might be looking at a single organism. But one is doing a database search where the total search space is 30 bits. In other words, the product of the length of the query sequence and the length of the database and residues is about 2 to the 30th, or approximately 1 billion in all size. So if one needs 30 bits to rise above background noise, and one knows on average how much information you get per position, you can calculate how long an alignment needs to be in order, on average, to get above this background noise. I simply divide the relative entropy into 30. So on this axis here, I am giving the critical length in bits, the critical length, assuming 30 bits, the length is in residues, by simply dividing the relative entropy into 30. So when I get out at 1 bit per position, the critical length is 30. What you'll see, and again, this should make beautiful sense, is that the more distantly related sequences are, the longer the alignments need to be, on average, to get enough of a score to rise above background noise. If you have sequences that are virtually identical, the critical length here can be very short. Here, if I divide 4 into 30, I get about 7 or 8. And so the critical length here is 7 or 8. If I get an alignment much longer than that that is identical, that's unlikely to occur in a database search of this size purely by chance. In fact, a number of years ago, there was a short letter to Science and a number of letters in reply that occurs here. This was back in '91. These researchers took a short sequence, glutamic acid, lysine, valine, isoleucine, serine, which in the one-letter code is Elvis. And they searched the database with this and in fact found Elvis occurred many times. We would not be surprised by this because this is shorter than length 7. If instead of using Elvis, they had, say, used the peptide that spelled out Springsteen, for instance, if they decided to search for the boss rather than the king, it would be very unlikely that this would have occurred. But going back here, as you get very closely related sequences of more than about 8, 10 residues, it's very unlikely that you will find them purely by chance. Now, I've left one thing out in this discussion, and that is that this graph is assuming that we know the distance with which the sequences we're trying to align are diverged. In other words, this graph is assuming we're using a 0 PAM matrix to compare sequences that have diverged by 0 PAMs. We're using a 100 PAM matrix to look at sequences that have diverged by 100 PAMs, etc. However, in general, we don't do this. We use one matrix for the database search, and we use that matrix for all evolutionary distances. Now, what happens if we use the wrong matrix for a given alignment? Suppose I'm comparing two sequences that have diverged by 120 PAMs, that have diverged by 100 PAMs, but I'm using the 120 PAM matrix. Well, I want to introduce a new concept here, which I'm going to call the efficiency of a matrix, and the graph of the efficiency of various matrices occurs here. This is taken from this paper. Each of these curves represents the efficiency for a particular matrix. So this curve represents the efficiency for the PAM 120 matrix, and this curve represents the efficiency for the PAM 180 matrix. The way to think about these curves is saying, if I'm using the PAM 120 matrix, but I'm comparing sequences whose actual PAM distance is not 120 but something else, how much of the information I could have gotten by using the right matrix do I actually get by using the wrong matrix? In other words, if I am not at 120 PAMs exactly, but somewhat off of that, but I'm using the 120 matrix, I will lose some of the available information. And you can see that the farther I am away from the right matrix, the more information I lose. And so the efficiency drops off. Now, you may wonder, well, how efficient is it important to be? And this will depend to a certain degree on how much information there is in the alignment. And this question also tells us why one tends to use a particular sort of matrix in a database search. Imagine that the alignment that we have actually has, if we use the right matrix, suppose the alignment has 100 bits of information. So if I use the right matrix for this particular alignment, I have 100 bits of information. Now, if I use the wrong matrix and throw out half of that information, so I'm getting only 50 bits, it's still going to stick out like a sore thumb in a database search, and I will find that alignment. And so for very closely related sequences, or for sequences that are close and long, so they have a lot of information, it doesn't really matter which matrix I use because I will find it even if I'm throwing out most of the information. On the other hand, suppose that if I use just the right matrix, my alignment has only 20 bits of information. Well, no, I will miss this alignment no matter what. It will be way down in the noise. I will not be able to distinguish this alignment by chance. And so alignments that have very little total information, again, it doesn't really matter which matrix I use because I will miss this alignment no matter what. Where it matters is when the alignment has just about enough information to be found in a database search. These alignments have come to be called in this field alignments from the twilight zone. This is a reference, those who are not aware, of an old television program from when I was a kid. But twilight zone alignments are ones that simply have just about enough information to be found by chance. And for those alignments, it is important to use the right matrix. Now, it turns out that for most sequences, if you have PAM sequences that are sequences whose degree of divergence is less than about 100 PAMs, they tend to have lots of information and be very easy to find. So it doesn't really matter what scoring system you use for these because you will tend to find them. If you are looking for alignments that have up here around over 250, 300 PAMs, then those frequently don't have enough information to be found in a database search no matter what you do. And it tends to be in this middle zone of sequences that are diverged by somewhere in here that you have difficulty finding these alignments. These tend to be where the twilight zone sequences are found. And so for database searches, the usual matrix that one wants to use is something like this. This is around the PAM 180, and this is roughly what the BLOSUM 62 matrix is roughly equivalent to that, has roughly the same relative entropy, and that's why that matrix tends to be the default for database searches. This paper suggests that if you really want to be very careful, you can use more than one matrix and do multiple database searches looking for very short but closely related sequences, middling ones and more distant ones by using different scoring systems. But usually using just this one will focus on the alignments that are the hardest ones to find. Now let me say a few words about DNA alignments. This whole idea of substitution scores and PAM matrices can be moved over to the DNA realm. If you have a very simple model of DNA evolution where you simply have a probability that a given nucleotide mutates and it has the same probability of mutating into any of the other 3 nucleotides, then when you work through the math, you find that you end up with match-mismatch scores. And so the PAM distance, this is now a nucleotide PAM distance. It means 1 change per 100 nucleotides. This is for a given nucleotide PAM distance, this is the percent conserved. So if you have 1 change, you tend to have 99% conserved. By the time I get out to 80 changes, I'm down about 50% conserved. This is the match score that you get in bits. This is the mismatch score. This is the score ratio, or the absolute value of the score ratio of the mismatch score to the match score. And this is the relative entropy in bits. So one can calculate exactly what the scoring system should be for any given nucleotide PAM distance and these are the percent conservation. So again, if you are looking for things that are a certain percent diverged, this table will tell you what match and mismatch scores would be optimal for that. Now, one can make a more complex model in which transitions and transversions have different probabilities. A transition is a mutation that changes a purine to a purine, in other words, A to G or vice versa, or a pyrimidine to a pyrimidine, in other words, C to T. Under certain models of the relative frequency of transitions and transversions, one comes up with a slightly more complex scoring system in which matches have different scores depending on, mismatches have different scores depending on whether the mismatches are transition mismatches or transversion mismatches. And one can construct a table like this for this more complex model, and if you want, you can look it up in this paper. Now, this allows us actually to ask another question that arises sometimes, and that is, if I have two DNA sequences, but the DNA sequences are both coding for protein, is it better to compare the DNA sequences directly, or is it better to translate the DNA sequences into protein sequences and compare the encoded proteins? Now, you will often hear the rule of thumb that it is better to translate into proteins and compare the proteins, and people said that somehow the proteins had more information. This always bothered me because the proteins don't have more information. In fact, all the information is there in the DNA. You've thrown some away in order to translate it into the proteins. So why would it be better to compare the proteins? Basically, this has been a shorthand, and the shorthand is that to compare the DNA sequences properly, one wants to understand the genetic code and look not just at the individual letters, but look at the codons that they encode, because, again, evolution is going to select out certain mutations that change one amino acid into an amino acid that has very different properties. So the sensitive way of comparing two DNA sequences to use the genetic code, but what that really basically is saying is translate it into proteins. You can milk some, perhaps a little bit more information by looking at the individual codons and coming up with a 64 by 64 codon versus codon table. But most of that information is there by translating it into the proteins. Now, to do this comparison, what I've tried to do is look at a DNA PAM distance and try to see what this means as a protein PAM distance. This is a little complicated for two reasons. First of all, one change per 100 nucleotides is really one change per 33 codons. And so if the change turns into an amino acid change, one PAM at the DNA level is actually more like 3.3 PAMs at the protein level. But there's a problem in that many of the DNA changes are silent. They don't translate into changes at the protein level. And so you sort of have to look at some data to see the relative frequency of silent versus meaningful mutations at the DNA level. And if you do this, you can come up with this rough equivalence of 24 DNA PAMs being roughly equivalent to 30 protein PAMs. So one can find out the information per residue in bits. One can look at the information per codon in bits because one's really these are the equivalent things one's comparing. At the DNA level, one has a much longer alignment. One's comparing codons. And when you actually find that there is more information at the at the DNA level when you are at very close distances. In other words, at 0 PAMs, we said you have about 4 bits per position at the protein level. At the DNA level, that's 3 identical amino acids, each of which gives you 2 bits, so you get 6 bits of information. If, however, you are using scoring systems like the ones I've described before, this information decays much more quickly. And just around 50 DNA PAMs, you have a crossover point. And out here at 150 PAMs, you are still getting 3/4 of a bit per amino acid, but you've thrown out almost half of that information at the naive DNA level, where you're simply looking at individual DNA changes. And so an alignment like this may be very easy to find at the protein level, but completely impossible to find at the DNA level because you have thrown out more than half of the available information. So we'll go just about 5 more minutes because we got a slightly late start here, and say that we have talked here about what are the optimal scoring systems to use for comparing protein sequences, for comparing DNA sequences. Sometimes people forget that we are dealing here in a very restricted context, and that is comparing 2 sequences where we take nothing into account except which amino acids align with one another. There is no thought of any kind of correlation. One is dealing with pairwise alignments. In other words, we've talked about optimal scoring systems in a very restricted context. And there is a lot more information potentially available if one expands the context. Going beyond pairwise sequence comparison, one simple way to get more information is that if you align a lot of sequences, you may find that in a given position where you might have a leucine in the first sequence, and another position farther down the protein, you also have a leucine. But when you align this with, say, 10 sequences, it may be that the first leucine is always a leucine in all of these 10 sequences, where the second one seems to be able to be anything. It may be glutamic acid in some, and cysteine and proline. The protein may not care what that amino acid is. You only learn this from the multiple alignment, and this allows you to adapt your scores to each position separately. So you have different scores for different positions, which you wouldn't know to use if you had just the one sequence. This allows you to construct position-specific scoring matrices, which are very closely related to hidden Markov models, which also have position-specific scores, but build them into a larger context of probabilities for insertions and deletions. There are various database search programs that have been built on these generalizations of pairwise sequence comparison. One that we developed here is SciBlast. Other popular programs are Hammer and SAM, which are both based on hidden Markov models. And these sorts of programs can run almost as quickly as the straight pairwise sequence comparison. One other way to get more information is to understand that proteins, of course, are three-dimensional objects that fold up in a complex way, and that frequently a position in one part of the protein will be in close proximity, spatial proximity, to a position in a very different part of the protein. the amino acids that occur at those two positions will be correlated because they interact with one another. And so if one can understand these correlations, one can make the score that one assigns to a particular amino acid at one position in the protein depend on the amino acid that occurs at a different position in the protein. These sorts of correlations can be inferred from a three-dimensional structure. If one has one for a protein, one can see which positions are near to one another, or you can sometimes infer these correlations also from multiple alignments. The problem with using these scores for correlations is that it makes the algorithms much more difficult. One of the great advantages of the dynamic programming algorithms that we outlined at the very beginning of the talk yesterday was that the score at a given position depends only on what you're aligning there. It doesn't matter what you're aligning somewhere else. If you don't know the score for aligning one pair of residues until you know the score for aligning a completely different pair of residues that occur later, the algorithms become much more complicated and take a very long time. And so although these threading algorithms can be a lot more sensitive than pairwise substitution, they also take a lot more time. Whereas these position-specific scores that still ignore the correlations between different positions can capture a lot more information without being slowed down substantially. So that is the end of today's talk. To the extent that there are any questions that have come in, I will try to answer them.
So, yeah, there's a question about polarity bias from the audience.
Okay, so I guess what you're referring to here are different amino acids. Some may be polar, some may be nonpolar, some may be hydrophobic, some may be hydrophilic. All of this is basic. When people started constructing scoring matrices without having the insight of target frequencies, one way that they constructed these matrices, in fact, were to look at polar, nonpolar residues and come up with different properties like this, size, polarity, other properties, and try to explicitly build these into the score. By looking at the data that Dayhoff did, by looking at what actually tends to mutate into what and get accepted by evolution, or looking at more distant relationships, these polar relationships, hydrophobicity, and so forth, are all very evident in the scores that occur. So that one finds by looking at these matrices, if you go back to last week's talk where I actually showed you the BLOSUM 62 matrix, you will see that similar amino acids, for instance, arginine and lysine, which both tend to be positively charged, will actually have a positive score, whereas, for instance, aromatic residues such as tryptophan or tyrosine will also tend to have a positive score. This comes very naturally out of what evolution allows to take place. And so one doesn't have to worry explicitly about taking account of these things. They are taken account of by the frequency with which various amino acids tend to align with one another.
All right. Some of the other sort of more minor questions we'll go ahead and respond to offline. And then if you want to send us an email at webinars@ncbi.nlm.nih.gov, we will try to get those answered as well. In answer to the most common question, we will be posting the video of this webinar online, most likely within the week. And also the video of Stephen's webinar from last week is online. If you missed it, it's excellent, and I encourage you to go watch it. That said, I'd like to thank Stephen for the webinar. I certainly learned quite a bit from the last part of it, and I hope you all did too. And have a nice day and a nice week, and we'll look forward to seeing you in future webinars. Thank you.